Hi everyone. In this lesson, we're going to go over antibody-mediated specific immunity, which is also known as humoral immunity. In this lesson, we'll cover the parts of your textbook that specifically relate to antibody-based immunity. So, in this process, we're going to learn all these vocabulary words and we're going to cover these topics. Okay, so just to remind you about specific immunity, also known as acquired immunity, also known as adaptive immunity. These are specific defenses that are created or done by lymphocytes. Lymphocytes are special kinds of white blood cells that specialize in attacking an antigen. So any given B cell or any given T cell, it is only able to attack one antigen, but any pathogen that has that antigen that T cell or B cell is specialized for is going to be nuked by those things. And antibodies are special proteins that are created by B cells to help assist with those attacks. Sometimes those antibodies are also called immunoglobulins. The thing that makes them unique to other white blood cells is because they're specific to the antigen that they respond to. So a T cell the T cell for a given pathogen will only attack that given pathogen, or only that given toxin, or only that given cancerous cell type. Okay? Now, just a quick heads up. When we talk about specific immunity, that doesn't mean the other parts of your immune system are gone. So we have innate immune system and then specific immunity. What actually happens is when the specific immunity occurs, it's because the pathogen has bypassed your innate immune system. But that doesn't mean those immune parts disappear. In fact, a lot of the time, your specific immune system works with your innate immune system, especially the white blood cells involved in your innate immune system, like macrophages, and they actually enhance their function in addition to those B cells and T cells doing their thing in specific defenses. Now, I talk about specific defenses as things that respond to antigens. So just to remind you what antigens means, those are substances the immune system recognizes as non-self. So those could be pathogens, those could be viruses, those could be bacteria, but they can also be non-pathogens like pollen. Anything someone is allergic to is an antigen to that person because their body recognizes it as non-self. And one of the other issues that we find when it comes to the immune system is transplants. Sometimes people's organs fails and you want to replace it. But one of the issues with that is the person's body will now recognize that incoming organ as not themselves, and they'll attack it like it's a virus and potentially destroy it. So a lot of people who have transplants have to take drugs that basically suppress and weaken their immune system in order to prevent their immune system from destroying their transplanted heart, for instance. And this is all a byproduct of a really key thing that your body does, which is called self-tolerance. It's impossible for you to specifically just design B cells and T cells against a virus as it comes in. It's really hard for your body to do that. So instead, what they do is they make millions, if not billions, of different types of B and T cells, and then they remove all the B and T cells that react with your own body. And the only ones left remain are these million to billion different types of B cells and T cells that could respond to any potential antigen. So even if a virus that your body has never seen suddenly comes into your body, a B cell and T cell will eventually react to it. Not immediately, but eventually. Okay? Now, when we talk about that ability to distinguish self from non-self is a really big deal, and actually part of why your immune system is so flexible against so many different things, so many different bacteria and potential viruses. Now, when we talk about B cells and T cells, they respond to the location of antigens, the location of potential pathogens in different parts of your body. So B cells are actually good about finding invaders outside your cells, finding antigens floating outside the cells before they've entered a cell, for instance. But if they've entered a cell, they might be able to evade those B cells. So then we have the T cell to help us recognize if an antigen is being produced inside the cell. If a virus has snuck into a cell and taken over that cell, then it's capable of discovering that. It's also good at finding out if a cell suddenly has turned weird on you, like become cancerous. And a part of that is our cells have to display contents inside of them to help your body identify any infected cells. We'll talk about that more in another lecture. All right, so this is sort of the display mechanism. All cells are required to not just mark themselves as self, but to also like display random stuff inside of it so that no infected cells can like sneak past your immune system. They're forced to like at least show a glimpse of what's happening inside, which could be virus production. So those white blood cells that are really critical to specific immunity, we call those lymphocytes. And lymphocytes, just to remind you, are a special group of white blood cells that are core to adaptive immunity. They allow for all the key parts of adaptive immunity, which includes self and non-self recognition to avoid attacking your own tissues, specificity and diversity, the ability to specifically attack a given antigen, and the diversity to attack for your body to have the ability to attack potentially millions, if not billions, of different types of antigens. And memory. If you have responded to an antigen for a pathogen, then you're going to be able to respond to that same pathogen in the future in a much stronger and much faster way. Your specific immune system usually takes a week or longer to get kicked into gear and to get engaged. But once it is engaged, once that you recover from the illness, if you see that virus again in the future, you'll actually attack it immediately rather than taking a week longer because of that lingering memory. All right, so the groups of lymphocytes that we're going to cover are B cells, T cells, and natural killer cells. B cells and T cells are the main group we're going to focus on. B cells produce antibodies that are key to humoral immunity, which is responding to infection that is occurring outside of cells. Bacteria and viruses that haven't gotten into a cell but are floating around in your blood or fluid just around your tissues. And just a heads up, if you're in anatomy, it's really important that you know that B cells mature in the bone marrow. So all white blood cells are made in the bone marrow, but B cells do their final stage of maturity before they become active in your bone marrow. T cells are central to the response against infected cells or cancer cells. So they spot when the inside of your cell is doing bad stuff. And they also play a big role in regulating your immune system. And we'll get a lot more into how T cells work in the next lecture. So these guys are really core to what we know as cellular immunity, which is detecting and fighting off cells that are doing something wrong inside of themselves. All right. And if you're in anatomy, it's really important that you know that T cells actually are formed in the bone marrow, but then they go into a thymus, which is a special organ above your heart, where they mature there. And last but not least, the last group of lymphocytes we have are natural killer cells, which help fight cancer, but they're not really critical to specific immunity like we're going to be talking about. So we're not going to really talk about them too much after this point. We're going to really focus on B cells and T cells. Today, we're going to really focus on B cells, though, because those are core to humoral immunity. Now, humoral immunity, by the way, is actually originated from the word humor, which is a medieval term for all the fluids in your body. You may have heard of bloodletting done in medieval times to treat illness. And that's part of the reason because they used to think the inside of your body was based on the balance of different humors, like red humor, black humor, yellow humor, to like refer to bile and blood and urine and stuff like that. And they would think that because your humors were unbalanced, they had to remove some or add some into your body as a treatment. And that's why treatment by doctors in the medieval times was literally sometimes a crapshoot. But anyways, these guys are antibody-based immune systems that defend against pathogens as they're circulating around your body, as they're heading for your cells, or not necessarily heading for your cells, but floating around your body. All right? Now, B cells can be considered into two groups. You have plasma cells, which are kind of effector cells, and memory cells. So plasma cells are the active B cells that are actively making the antibodies you need. And they're considered an effector cell. An effector cell just means the cell that's actively responding to the situation. All right, you have different types of effector cells in the T cell category, and the effector cell in the B cell category is plasma cells. And then you have memory cells. You have memory B cells and memory T cells. Both of them are just the specific, if you activate a B cell to become a plasma cell, you'll also create a memory B cell for that same antigen you're using in the plasma cell, so that that memory B cell will linger and stay in your body and stay trained in your body in the event you get reinfected by that same pathogen. Then, because it's still present, it will respond much more quickly to that virus. And instead of taking a week to respond, it'll immediately respond and block that virus. This is what we call immunity. When you're immune to a virus, it's because you have lots of memory cells in your body, memory B cells, memory T cells, against that virus. Okay, so kind of to visualize this, if you have if you get an infection, generally speaking, you have a B cell that's producing antibodies, and you have some memory B cells lingering, and those memory B cells can rapidly convert into plasma cells in the event of a reinfection and quickly produce antibodies in the future. And you'll also have, after an infection is over, a few B cells lingering, plasma cells lingering, to keep pumping out some extra antibodies, at least for the next few months, if not longer than that, to have a little bit of just extra added protection or in your body after infection is over. Okay, so this whole B cell response of an invader coming to your body, and then the B cells in your body test against that invader, and any B cells that particularly bind to an invader, those are the ones that are going to be recognized and then cloned really quickly, and they're going to grow very quickly, rapidly produce new cells, and then turn into either plasma cells to make antibodies to actively fight the virus, or convert into memory cells. In fact, both of these occur at the same time. But this whole B cell response takes a week, 2 weeks to really kick into gear. So that's why oftentimes antibody tests don't work on someone who's currently infected with COVID, and you only see those antibodies present in their body after they've been infected with COVID for a couple weeks, or after they recovered from COVID, before those antibody tests actually work on them. Antibodies, just to let you know, are Y-shaped proteins that bind specific antigens. So generally speaking, when people hear of you being immune to something, they usually go, oh, it's because you have lots of antibodies against it. Antibodies are kind of famous in terms of how they're associated with immunity. Okay, so these are multi-chain proteins made by B cells, and each antibody is unique and specific to the antigen they're making. So a B cell that makes one kind of antigen only makes antibodies against that antigen. All right. IG is sometimes the short name that we give antibodies for immunoglobulin. And they're going to bind to molecular shapes of antigens, and they're going to be able to respond to, you're going to make millions in your body because you have millions of different B cells. You're also going to make millions potentially of different antibodies to respond to particular antigens. Okay? And we consider these tagging handcuffs on foreign substances. So if you have an antibody that's capable of binding to an antigen, it's going to cling on to it and really ruin anything with that antigen's day. So a pathogen, a virus that's holding a particular antigen, if you can get an antibody to cling on to that virus, it's going to prevent that virus from infecting, and it's also going to help signal to your body to scoop up that virus and destroy it. Okay, so those binding regions on the antibodies are really key for the antibody's ability to really destroy and hinder a pathogen that's floating outside your cells. Okay, so B cells has actually tons of antigen receptors on its surface to help do that whole process. Don't worry too much about this. Just really focus on the fact that that binding region really is critical to the antibody's ability to do its job. So that Y shape is a very simplified form of the actual structure of the antibody. It's quite complex, but if you distill it down, you always have a kind of region on each end of the antibodies that's capable of binding the antigen that you're trying to. And this specific antigen is only going to bind one particular antigen and not any other. So you have to make a different antibody if you want to bind something else. That's why they've talked a lot about variants for COVID, because if COVID change alters too much, mutates too much, you're going to need a different antibody to cling on to a potentially altered COVID virus. Because B cells produce a specific antibody that only binds a particular antigen. So these B cells, you can see, only bind a certain shape. But notice here for this virus, it can respond to 2 different antibodies in this list because the same pathogen, the same virus, contains 2 different antigens. Viruses and bacteria have, generally speaking, hundreds, if not thousands, of different antigens on their surface. But as long as you make an antibody against at least one of them, you can attack it. So if you can teach your body through vaccines, for instance, how to attack one specific type of antigen, you can grab onto that virus or bacteria as long as it's holding that antigen. Okay, so how the antibodies actually do their job. They play a major role in what we call neutralizing pathogens, stopping them. And because antibodies are in your blood and your body fluids, they help. If you have a good presence of lots of antibodies, they'll actually grab onto a virus or bacteria before they have a chance to get into a cell or destroy your body. All right, so this ability to grab on and stick to a pathogen, it can sometimes lead to something called agglutination, which is when you actually have chains, one antibody or several antibodies create clumps and chains of multiple bacteria or viruses stuck together. And it actually helps your body scoop out those viruses or bacteria even faster. And then they'll also do other things. If they cling onto a virus or bacteria, that will actually often help signal to white blood cells to go ahead, your phagocytes, to go ahead and eat that material. And it can also help induce functions by complement, which is that protein in your innate system that helps destroy things, and also actions by other lymphocytes like T cells to destroy that material, destroy a bacteria cell, for instance. And they also help lead to signals that actually release cytokines to actually kickstart your immune system and make it even a little bit stronger in those white blood cells that are helping out and attacking things based on antibodies. Okay, and this is just a different image of what I had on the last slide if you want to take a picture of it or refer to it for your notes. Okay, now antibodies, there's actually a lot of different types of antibodies. We're really going to focus in on 2 kinds though. One is called IgM and one is called IgG. So immunoglobulin M, immunoglobulin G. So the M antibody, IgM, is actually the one that's usually produced first in your immune system response. And it really actually initiates and helps activate complement proteins, reminding you complement proteins are proteins that help that are antimicrobial and help destroy and take down bacteria and viruses to an extent, but they're really good at taking down bacteria. All right. Now, this is usually the first kind of antibody created. Then, if your immune system is still going, the second response or the later responses of your immune system will start creating lots of what we call IgG, which is an antibody that's like really good at taking over and circulating your blood and destroying things. And they're good about tagging things for phagocytosis, for destruction by white blood cells that are eating it. Both of these are really critical to a disease. Generally speaking, the beginning of your illness, you got a lot of IgM, and towards the end, you have a lot of IgG. And after you recovered from the illness, you still have a decent amount of IgG left in your body to help prevent that virus from re-entering. So when you're taking antibody tests, they actually detect both IgM and IgG, but they're actually much better at detecting IgG antibodies. Those are the ones you really want if you've recovered and are trying to prevent a virus from coming back in. All right. So you also have three other kinds of antibodies that are often discussed, but we're not going to go super into them, so don't worry about it so much. But if you're curious, we have IgA, IgE, and IgD. IgA is the one that mothers can give their babies through feeding them breast milk, and IgE is the one that is associated with like allergies and stuff. So remember, with those antibodies, it usually takes a week, if not a week, for them to fully develop, and even longer than that if you want IgG antibodies. And a lot of your antibody tests that you've heard about when it comes to COVID, they're really more useful for testing if someone has been infected with COVID because they're good at detecting IgG better than IgM. And again, IgG takes a couple weeks for them to really peak and linger in your blood. But they're also a little bit unpredictable as tests because for people with COVID, that antibody we found has dropped off after a few weeks, and it really varies person to person how much antibodies you have lingering in your blood after infection. We also have a different test. It's not the antibody test, it's a test for RNA virus genome that tests if you're presently infected. And they're seeking for the presence of RNA due to an active infection because those RNAs are found in the viruses are infected. And this one is decent, though it also, not only is it for present infections, but it also is for present infection in the tissues they're swabbing. So some people, unfortunately, don't always express the virus in their nose or back of their throat, so not all RNA tests, virus tests will work on them. So just a heads up, just because you get tested to see if you have COVID and it says no, that doesn't necessarily guarantee you don't have COVID. It definitely means you have a lower chance of it, but there are people that will sometimes not express any COVID virus RNA in their nose or back of their throat and will instead express in their lung, and then this test would actually show them being negative even with a COVID infection. So just a heads up. Both of these tests are pretty good. They're like sometimes 90% plus, not always, but they aren't perfect. So just a heads up. Okay. Now, another thing I wanted to mention is your T cells and B cells responses are generally what leads to immunity, especially the production of those memory T cells and B cells post-infection. And generally speaking, your antibodies after infection will stay, but then eventually they'll drop off. Sort of like if you train someone to do something, but they don't use that skill afterwards, they'll forget it eventually. However, if you are exposed to that same antigen, that same bacteria or virus a second time, your immune system, your lingering memory cells, rapidly fight it off very quickly. That's what we consider immunity. And they'll retain those antibodies and retain even more memory B cells and T cells afterwards, so that after that second exposure, you're just really good and very strongly able to fight off that disease and even prevent any effect from it a second time, or not even a second time, a third time if it comes back. That's what produces long-lasting immunity. So this is a thing we're aiming for vaccines. So just think about it. Like if you're trained to do something but you don't use it ever after the training, you'll forget it. But if you've trained in something and you actually actively have to use it and practice it, then it's going to stick around for longer. Your immune system does something very similar to that. Okay? Now, we usually talk about antibodies when we talk about your immune system and your immunity against a disease. And oftentimes when we want to vaccinate, we're trying to produce this second immune response with that lingering antibody afterwards. But I just want to point out that these, even though I'm saying antibody concentrations, it's usually used as a proxy because antibodies are the easiest thing to measure. But along with those antibodies lingering, that also usually indicates a good amount of B cells and T cells also lingering, those memory B cells and T cells being present and kind of in the background. So just a heads up, antibodies are usually used as a proxy to measure how well your body may respond to future encounters against the pathogens, but it's actually your B cells and T cells are just as, if not more important than the amount of antibodies in your blood. They're just harder to measure. So usually that's not the easiest way of measuring if a vaccine is successful. They just mainly pay attention to the antibodies. All right. Now, when it comes to the antibodies, there's two ways to gain those specific immunity by B cells and T cells. We have active immunity and passive immunity. Active immunity is what we want, generally speaking, because it's the long-lasting immunity that you're going to get from either recovering from a virus or disease or getting a vaccine. All right. Passive immunity is good for temporary immunity through medicine, or if you're a baby, through drinking mom's breast milk. All right, this stuff is temporary and short-lived. It's putting antibodies into someone's body without putting the B cells that are producing the antibodies. But when it actually, so you might have heard of like antibody infusions into people with COVID, and those are okay for immediate treatment of serious COVID-19, but we're actually finding out it's not as effective as other drugs. But it has been a technique people have used. But really, we're finding the most effective way of preventing and getting rid of COVID is getting those long-lasting B cells and T cells in people's body through vaccines. And that's what we were talking about when we're talking about vaccines. Now, we sometimes call the process of getting a vaccine immunization. So there we go. Those are procedures used to induce immunity. There's different types of procedures, but the main one we use in modern times is vaccines. So in this case, we're going to elicit immunity to a specific antigen or a specific pathogen by stimulating the body to produce B cells and T cells against it, by exposing the body to some kind of antigen that's normally found on that pathogen. You might expose them to a weak version or harmless version of the antigen from that given virus or bacteria. Vaccines aim to expose the body to antigens of a pathogen without allowing the disease. So there's different strategies for vaccines. You can just don't even give them the whole virus or bacteria. You just give them parts of it, like a protein that's always found on a virus, but not the actual virus itself. So sometimes the vaccine will consist of just the protein itself, like you'll make the protein and purify it and put it into the person's body, or you can give the person a small little RNA or DNA and have their cells produce the protein piece that you want the vaccine, you want them to protect against. A lot of the modern vaccines that we've been talking about use mRNA or DNA to do that. And so instead of actually giving you the protein in purified form, they teach your body to produce the protein, and then your body learns to protect you against that protein. Or they can just directly inject a weaker version or a killed version of the virus into your body, though there is a little bit of risk to that sometimes, because that's not going to stimulate all parts of your immune system. Because this virus isn't going into your cell, it's only killed and whole. You only stimulate humoral immunity, and maybe not, you won't stimulate as good cellular immunity against it because you're not showing your body what the antigen looks like inside cells. All right, and if you're curious about it, you can look more into the different types of viruses that are summed up in this chart here. And just to put things in perspective, with today's modern vaccines, and especially RNA vaccines, it actually usually takes like 10 years for a vaccine to fully develop. Prior to this year, I think the shortest vaccine that was ever developed, it took 3 years. This vaccine, our vaccine, we've made it and sent it to market, have pushed it to millions of people within a year, which, as sad as the news has been and how depressing COVID has been, our vaccine game has been strong. It has been really strong, surprisingly strong. Okay, so anyways, put it into perspective, this summary goes over kind of the key parts of what we covered in our lecture, including the difference between cellular and humoral immunity, as well as the B cells' major role in humoral immunity and how vaccines take advantage and help stimulate specific immunity. Bye.